so on, and eventually we'll actually start talking about microbiology. All of this has really just been a prologue, introduction, and so on. You really needed the chemistry, and then parts of the cell you also need. And I'm sure you've had a lot of it already. So we'll start off with the cell membrane, and it's called a lipid bilayer. Bilayer means a double layer. And it kind of looks like this. That's your bilayer. And those two strands coming down, think back on phospholipids. It was two fatty acids, and then a phosphate, and then the glycerol. So these two strands are your two fatty acids in the phospholipid. I'll show you a picture. Don't look at it too much because you might go blind. See, really blurry. But you can still even so see what I have there, the double thing there, phospholipid. And that's enough because you might go blind. I don't know why it came out so blurry, but it did. But at any rate, that's the lipid bilayer. All membranes look like that. They've all got this type of structure. And they're characterized by the fluid mosaic model. The fluid mosaic model. Now, many of you already know what a mosaic is. It's it's a picture made up of little colored stones. And like they're on the wall. So you get really close, like that. Well, all you see are some colored stones. If you back off a little bit, you see a picture. And that's a mosaic. Well, the membrane is like that. I've been saying it's made out of phospholipid, and it is, but that doesn't mean that's all there is. There are other things. You do have proteins in there, you do have other lipids, you do have carbohydrates. But also, you have combinations. The main combination is glycoproteins. Glycoproteins. Now the glyco means sugar. And then protein. So these are sugars bonded to proteins. They're extremely common. And we'll be seeing these all quarter long. The glycoproteins have two main functions. One is information molecules. You know, different parts of the body need to know what's going on in other parts of the body. Glycoproteins are often the messengers that will carry information from one part to another. I can tell you how it works. It's off the record again. But let's take glucose, for example. This time you should really recognize glucose. So, I think many of you are probably aware that computers work on a binary code. That basically all the computer information is just a bunch of zeros and ones. But the computer, of course, can read it, well, slightly faster than we can. Well, carbohydrates work on a binary code. So for example, the hydroxyl here is pointing downwards. That can be given a 0 value. And if it points up, it can be given a 1 value. So here, 0, 0, 1, 0. That would be a binary code for glucose. Carbohydrates carry information by binary code, just like computers do. And they act as information molecules in the same way. Anyhow, off the record, so you don't need that, but I wanted to show you how it does work. So all we care about, back on the record now, all we care about is information molecules. Also, two functions, also for stickiness or adhesion. Glycoproteins can be sticky. So a lot of times glycoproteins are used to kind of stick two cells together or maybe two molecules together. So two main functions for glycoproteins: information molecules or stickiness, adhesion. We can have other combinations. Glycolipids and lipoproteins. So those are your combinations.
That's in the fluid mosaic.
I'm sorry.
That's in the fluid mosaic model.
Well, it's in the membrane.
Oh,
The
in the
fluid
membrane.
mosaic model is only describing the membrane. Excuse me. The fluid mosaic model only describes the membrane. So the glycoproteins, etc., are all parts of the membrane. And the fluid mosaic model helps to describe that.
They all have the same functions. Those should the same function.
No, they have different functions. They have different functions. But the glycoproteins are by far the most important. So at any rate, you have all of these, and you have basic proteins, etc., that are all part of the membrane, and thus the mosaic. The membrane's like those pictures with lots of little colored stones. All the little colored stones make up a big picture. All the different molecular types make up the membrane. So those different molecules are like boats floating in the ocean. The phospholipid is the ocean. And those different molecules are boats floating in the ocean. So the phospholipid is still making up most of the membrane. There's just other stuff in there. It's not only phospholipid. The fluid part is because the membrane is not made out of concrete. The parts of the membrane can float around. It's not like water. It's not going to flow like water. But basically, the parts can move. So thus, the fluid mosaic model, you've got different parts, different pieces of the membrane that can move around. A little bit, not a lot. Now, some examples of how this works. Recognition proteins. Now, I think all of you know already, part of the function of a cell membrane is to keep things out. You don't want everything getting into the cell. But you still want some things getting in, like food, for example. So you do want some things getting in. The recognition proteins act like scouts. Their function is to determine if something should get in or should not get in. Basically asking friend or foe. And if the molecule, whatever is trying to get in, is a friend, then the recognition protein says okay, and it gives permission to the receptor protein. So the recognition protein is getting information, recognizing, identifying whatever's trying to get in. Based on that, it won't surprise you to know recognition proteins are glycoproteins. They're information molecules. So they're glycoproteins. And then, again, permission is given to the receptor protein. So the receptor protein, it's a receptor. So it will then grab onto whatever it is. It'll bind it. So that, again, now will not surprise you. Receptor proteins are glycoproteins. The first one is doing recognition, identification, information. Second one doing adhesion, stickiness. They're both glycoproteins. The receptor protein is going to grab onto whatever it is, and then we'll pass it on to the transport proteins. The fact is, it still needs to get through the membrane. The transport proteins can do one of three things. One is, it might just form a pathway. It might form a tunnel through the membrane. And basically, when the receptor protein grabs onto whatever it is, then the doors open up and the thing can just simply go through the tunnel and get to the other side. That's one possibility, that it acts like a tunnel or a pathway or a road or something. Second possibility, the transport protein might be a carrier molecule. It might act like a truck or a ferry and physically carry the particle through the membrane to the other side. So it might be a carrier of some sort. Third possibility, I don't actually know what to call this. The third possibility is the transport protein will grab onto the particle trying to get through. So this will be, part here, this will be your transport molecule. The black cap will be what's trying to be carried through. If you start out like this, then the transport molecule simply rotates. It rotates 180 degrees. So right now, the black cap's on the outside, now it's on the inside. I don't know what to call that. But anyway, that's another possibility. So basically, three different ways.
Is that a glycoprotein?
Yes, actually, yes, it is also a glycoprotein. So the first three of these are all glycoproteins. The fourth one is not. So the first three are all glycoproteins.
In doing the turn, what exactly does it do to the particle?
It doesn't do anything except it carries it. It physically is transporting it from the outside to the inside. It's purely a movement thing. So all three of those are glycoproteins. The fourth of these, channel proteins, those actually are for fluids. These first three are more or less carrying solid particles from one side to the other. The channel proteins form a canal. They form a channel, but they form a canal and the fluids can pass through the canal.
So it doesn't need sugar.
No, no, it's just plain protein, yeah, no sugar. Not a glycoprotein. Now, before I show you a picture of that, which will not make you go blind, just a couple more things to throw at you. One is the integral proteins. That's a general term. Integral proteins go all the way through the membrane. They go all the way through. You'll see a picture in a second. Peripheral proteins are only on one side or the other. They're only on one side or the other. So, for example, the recognition proteins will be peripheral. They're only going to try to recognize what's coming in. So they'll be peripheral. The receptor proteins would be peripheral. They're going to bind whatever it is that's on the outside. The channel proteins are definitely integral. They're going all the way through. The channel proteins go all the way through. The transport proteins could be either one. It might be peripheral, it might be integral. So the recognition proteins are definitely peripheral. Receptor proteins definitely peripheral. Channel proteins definitely integral. transport proteins could be either. Kind of depends. Take a look at a picture. So first off, you can see the lipid bilayer. That's typical of membranes. So that's phospholipids. See it here. That's phospholipid. See, here are your channel proteins. Those are integral going all the way through. There's your canal that allows fluids through. There would be a peripheral protein. Notice here a mistake. It's calling this an integral protein. It's not. It's only peripheral. This is integral, going all the way through, but this one is not. So even the big boys make mistakes. At any rate, that whole thing there, that's your mosaic. All these different pieces, all adding up to make your membrane.
Is this the picture that matches that?
I'm sorry.
I'm kind of trying to go through the way you lecture, trying to find the information in the book, but I don't know the match.
There isn't a chapter on it.
There is.
Where?
But the part we really want is the organelles. And we'll go there right now. So we don't need that part. So go to slide 3, which is the organelles. These are the parts of the cell.
So are you
That should
skipping that information that you just posted?
Pardon me?
Are you skipping that other information? That you
I'm only skipping
Yeah, that. Okay.
this.
Okay.
Wow, I do.
Christine, remember?
Okay.
I'm skipping that.
Okay.
What?
Oh,
I'm going now to organelles. The word organelle means cell organ. So the cells have organs just like we do. And we'll start off first with the nucleus. The nucleus. Now, of course, bacterial cells don't have a nucleus, but we do. So you still need to know what it is. And you all know this stuff. The nucleus is the brains of the cell. It runs the show. It monitors what goes in and what goes out.